If you look on the screen, there are 20 amino acids listed there, and those are the amino acids that our bodies use to make proteins. And if you think about all the types of proteins in our bodies, we have hair, muscles, nails, hormones, enzymes. Those are all different types of proteins, and they only use these 20 amino acids. So our body has to have a system where it can arrange those amino acids in the correct order so that your body knows which protein to make, because you don't want it to make the wrong protein at the wrong time. And that's what protein synthesis is all about. How does your body get the order right of those amino acids? It starts with the DNA. But before we get onto DNA and the whole thing of protein synthesis, we need to know about another important role player, RNA. And there are different types of RNA. So let's first have a look at the types of RNA. The first one you can see there is mRNA. It is messenger RNA, and it codes off the DNA in the nucleus, and it has codons. And if you look there, a codon is a triplet. There are 3 nitrogenous bases. They make up each codon. Another type is tRNA. Now, tRNA is known as transfer RNA. It has a job of transferring information. We'll get to that just now. It takes the amino acids to the ribosome, and it has anticodons. And if you look over there, you can see where they are positioned on the tRNA. The tRNA has got a funny shape. It's an odd shape. Another one that is maybe less important for you is ribosomal RNA, and you find that in the ribosomes. But let's get on to what we're going to talk about today. Here is a basic eukaryotic cell, and a eukaryotic cell always has membrane-bound nucleus with a cell membrane around the cytoplasm, and that is where protein synthesis takes place. Transcription, which is the first phase of protein synthesis, it takes place in the nucleus, and then translation in the cytoplasm, in that order. So everything starts in the nucleus. If you look over there on the screen, you can see 2 structures. The first one, they are both role players now in transcription. The first one is DNA. Let's just write it in there. And the other one is mRNA. Remember I told you just now about mRNA? That's messenger RNA. And those 2 are the actors or the role players in transcription. So let's see. First of all, what happens in transcription is the DNA unwinds, the hydrogen bonds break, and the DNA double helix unzips. Does that sound familiar? I'm sure you know what that's all about. That sounds similar to DNA replication, but you mustn't get it confused with DNA replication, because if you look at the next step, one strand becomes the template. Only one strand. So on one side of that, the template that's opened, you get, it provides the helix that's opened, you get the template, and that forms the code for the mRNA, how it must code. So free nucleotides form mRNA strand, so they come from all over in the nucleus, and they hook up next to each other depending on what the DNA is saying, and it forms little triplets of codons. I'm just going to show you over here. So if the DNA, for example, was G, C, let's say A, then the mRNA will be C, G, and remember it's RNA, so now the RNA, it doesn't have thymine, it has uracil. Then it will be CGU. And if you look at the triplet over here, that is CGU. So it means the DNA that it coded off was GCA. I just wanted you to see that. Then the next thing that happens is that the mRNA leaves the nucleus, it goes through the nuclear pore, and it goes into the cytoplasm. And that's the end of transcription. That's the end of transcription. And now we're going to start with translation. So there it's moved through. The mRNA, and you can now see, is at the ribosome, which is the site of protein synthesis. The organelle that where protein synthesis takes place is the ribosome. And I guarantee you're going to get that question somewhere in grade 12. They're going to ask you which organelle is involved with protein synthesis. It's the ribosome. Now we're going to look at translation, which is happening at the ribosome in the cytoplasm. The first thing is that each tRNA carries a specific amino acid. And you'll see that those little circles on the top of the tRNA, those are the amino acids. And each one has a different amino acid depending on what its anticodon is, because that hooks up with the codon of the mRNA. So let's have a look here. When the anticodon on the tRNA matches the mRNA codon, then the tRNA drops off the required amino acid at the ribosome. The amino acids, as they are dropped off, they join each other by means of peptide bonds. So they link up together, and between them, they are peptide bonds. Remember, the double helix of the DNA has got hydrogen bonds. But here, we're speaking about peptide bonds because we're going to form a polypeptide chain, which is a protein. And then a new protein is formed. Now let's have a look at the next slide. Here you can see you've got the tRNAs here. Here's an example of one. It's bringing the amino acid to the mRNA, and it is reading off the code on the mRNA. So let's say the mRNA was, let's say, here we can see it's CGU. Then if you look at what tRNA must come to that mRNA, it'll have to be G, C, and then U goes with A. And if you look here, there it is. So that one is going to come and it's bringing it to the mRNA and to the ribosome because of the mRNA's message that it's brought from the DNA. So it comes from the DNA to the mRNA. The mRNA then provides the codon, and the tRNA has got the anticodon. And because of the message that came from the mRNA, the tRNA will bring a certain amino acid. I just want to show you here that you've got what we call a polypeptide chain forming, and that polypeptide chain is what we call, it's another name for a protein, another way of expressing what a protein is, because it's a long string of amino acids joined by peptide bonds. And if you look at this last slide, I just want to show you, this is another diagram, because obviously I've got my own visuals, but there are lots of different ways these things can be asked or explained. And over here you've got the mRNA, and it's moving through the ribosome in that direction. And as it moves through, I'm just repeating what I've already told you, but I just want to show it in a different way. As it moves through, each little triplet here on the mRNA matches up with an anticodon on the tRNA, and there is the amino acid. So that is alanine. And if you think of that list I had in the beginning, alanine was one of those listed on that list. And now that one there is serine, and that's valine. So they're all different amino acids. And because of the code that came from the mRNA, that will tell the tRNA which amino acid to bring to the ribosome to make this lovely long polypeptide chain with peptide bonds in between the amino acids. Now, you also get asked questions with a genetic code table, but that's another video. So if you want to see how to use the table and answer questions about protein synthesis, then you'll have to watch that video. Just one last thing. The very important thing about tRNA is that once they've dropped off the amino acids, then, can you see this one over here? It's moving off, and it's going to go all over in the cytoplasm and look for another amino acid to pick up because of its anticodon that it's got, and it will bring it to the ribosome again. So the tRNA, think of them like taxis. They keep going off, picking up amino acids, coming back to the ribosome, picking up amino acids, back to the ribosome, dropping them off, going off again to pick up another one. I hope that's helped you. And if you enjoyed this video and it's helped you and you understand protein synthesis better now, why not subscribe now to my channel, and you'll get the next video and as they are posted.